Hello friends, how are you doing? And I hope you really all are having a great and fun time. So today we will discuss the questions of cell, the unit of life, and we will talk about the questions of CBSE on this chapter. Which organelle is responsible for increasing the surface area of absorption in a cell? Absolutely. The answer for this will be the endoplasmic reticulum. The endoplasmic reticulum, right? The endoplasmic reticulum is responsible, is responsible for increasing the surface area of absorption in a cell, right? So the endoplasmic reticulum is responsible for increasing the surface area for absorption in a cell. We know that in a cell what we have the nucleus in the center, which is surrounded by the nuclear membrane, right? So what we will see that there will be the nucleolus with the nucleoplasm inside and the presence of what? The presence of chromatic threads, right? And then what we will see that yes, these are the endoplasmic reticulum. So we see these how these endoplasmic reticulum are increasing the surface area of the entire cell. There will be some ribosomes attached sometimes, right? There will be some ribosomes attached sometimes, and sometimes these ribosomes won't be attached, right? So this is what? This is the endoplasmic reticulum, right? The ribosomes on this, and this is the whole of the nucleus. This is the nucleus, right? We move on to the next question. Define totipotency. Now what is totipotent cell? A totipotent cell is any vegetative cell that can grow completely into a new plant or a new organism, right? So a totipotent cell is a vegetative cell that can grow completely, right? That can grow completely into a new plant, and that is what is a totipotent cell, right? So totipotent cell is a vegetative cell that can grow completely into a new plant. We now talk about the next question, and that is name some cells in which the nuclei are absent, right? Name some cells in which the nuclei are absent. some of the cells in which nuclei are absent are so some of the cells in which nuclei are absent are number 1 the mammalian RBC, right, the mammalian RBC, mammalian RBC, and the second one, right, is the sieve tube cell in plants, the sieve tube cell in plants. Right, the mammalian RBC and the sieve tube cell in the plants. Now, what do we know or what do we think about the mammalian RBC? The mammalian RBC, we know that in a mature RBC of the mammalian cells of mammalian cells, right, the nucleus is absent. The nucleus is absent or the nuclei are absent, and the cells become biconcave, and the cells become biconcave disc shaped. Okay. So, in a mature RBC of mammalian cells, the nuclei are absent and the cells become biconcave disc shaped. So, what we see is somewhat like this. Because of the absence of nucleus in the center, what we see that this part becomes converges, or this part becomes narrow, and hence this is devoid of the nucleus. This is devoid of the nucleus. Now, sieve tube cells, we will see that there will be the presence of sieve pores, and these will be also, and there will be the companion cells on the sides and all, and also these will be lacking these sieve tube cells, right. They will be in continuation forming the sieve plate, and there will be the perforations also in these plates, and nucleus will be absent in these cells. Okay. So, then what will we see next? What are the differentiated cells, right? Here you need to understand 2 or 3 concepts which will be very important. First is differentiation, right? Then we talk about the de-differentiation. The fourth question we are doing, de-differentiation, and then we talk about the redifferentiation. Redifferentiation. Sorry. Okay. The differentiation, the de-differentiation, and the redifferentiation. Now, what do we say about the differentiation? A meristematic cell often divides and becomes differentiated, that is, there is a division of labor. For example, so what is happening, what we can write is a meristematic cell. What is a meristematic cell? A cell which is in the dividing state or keeps on dividing is called as the meristematic cell. So, meristematic cell gets differentiated into a specific cell for performing a specific function, right? For performing a specific function, right? A meristematic cell gets differentiated into a specific cell for performing a specific function. These are performing specific function, that is division of labor, that is division of labor. The dedifferentiation, what is happening? The differentiated cell gets back into the meristematic state, that is again it can divide. Okay, say for example a cell which was dividing, it was said, okay, you go and form the epidermis of the leaf. You form the epidermis of the leaf, you have to provide, functional, you have to provide the protective function now. So what happens in dedifferentiation, that function of protection by forming the epidermal cell is taken back, and it said no, you get back into the meristematic state, you divide again, and maybe you will perform a specific function after that. So dedifferentiation, here the cells, okay, here the cells are returned back into the dividing state, right? Here the cells are returned back into the dividing state, and this process is called as dedifferentiation, and this process is called as dedifferentiation. What about the redifferentiation? Here the cells again from the dedifferentiation they again get differentiated. Here the cells again get differentiated to perform a specific function, and this is called as redifferentiation, and this is called as redifferentiation. Okay, and this is called as redifferentiation. So now you know what are the differentiated cells. The differentiated cells are those, which are performing a specific structure, which are performing a specific function, or it may form a specific part of the structure of the plant or something. Name the two cell organelle other than the nucleus in which the DNA is found. Yes, the DNA is found in the nucleus. Are there any other organelle? Right. The two organelles in which the DNA is found are, right, in which the DNA is found are, one is the mitochondria, and second is the chloroplast. The mitochondria and the chloroplast. Now, because since the nucleus is present and these have the nucleus, right, the DNA, the DNA is present. The DNA is present in these cell organelles, so they can divide on their own, and hence these are also called as the semi-autonomous organelles, right? As these have the DNA, okay, as these have the DNA, they can replicate on their own, hence they can replicate on their own and are called, okay, and are called as semi-autonomous, organelles. As these have DNA, hence they can replicate on their own, and hence they are called as the semi-autonomous organelles, right? Now, what else? What are the dedifferentiated cells? I just explained you what is dedifferentiation and redifferentiation. So, what did we say about differentiation? We said differentiation is when when the meristematic cells get differentiated to perform a specific function, right? That is, there is division of labor, right? We said then that dedifferentiation, the cells which have already differentiated and performing a specific function, they get back into the meristematic state, they get back into the dividing state, back again, right? And that is dedifferentiation. And again, if they get specialized to perform a specific function, again if they want if they are there is the division of labor or the organization of labor, then we call it as the redifferentiation. Redifferentiation. So, differentiation, dedifferentiation, and the redifferentiation, right? Who proposed the cell theory and give its postulates? The cell theory was given by Schleiden and Schwann. Was given by Schleiden and Schwann. And was modified by Rudolf Virchow by the addition of another postulate, okay, and was modified by Rudolf Virchow, okay, and was modified by Rudolf Virchow by the addition of another postulate, right? The cell theory was given by Schleiden and Schwann and was modified by Rudolf Virchow by the addition of another postulate. So, what are the postulates of the cell theory? Number one, all plants and animals are made up of small units called as the cell. All plants and animals, right? all plants and animals are made up of small cellular are made up of cells. The cell is a structural and functional unit of all living organisms, right? The cell is the structural and functional unit, right? The cell is a structural and functional unit of all living organisms, of all living organisms, right? And thirdly, all cells arise from pre-existing cells. All cells arise from pre-existing cells, which is called as omnis cellulae cellula. Omnis cellula cellulae. Omnis cellula cellulae, that is, all cells arise from the pre-existing cells, and this last postulate was given by Rudolf Virchow. Okay. The cell theory was given by Schiller and Schwann and was modified by Rudolf Virchow by the addition of another postulate: all cells, all plants and animals are made up of cells. The cell is the structural and function unit of all living organisms. All cells arise from pre-existing cells, omnis cellula cellulae. Right. So this is the whole of the cell theory. We move on to the next question. Name one example for each of the following nitrogen fixing symbiotic bacteria, and this is Rhizobium leguminosarum. Okay, nitrogen fixing symbiotic bacteria, and this is Rhizobium leguminosarum. Okay, nitrogen fixing bacteria, leguminosarum, and a bacterium producing intestinal diseases man is E. coli. Bacterium producing intestinal diseases man is E. coli. Right, bacterial producing intestinal diseases man is E. coli. Right. Next, distinguish between the prokaryotic and the eukaryotic cells. Right. So, if this is the prokaryotic and this is the eukaryotic cells, what will we see? The eukaryotic cells. First of all, what we will see, the prokaryotic cells, these are not going to have a nuclear membrane. The nucleus is not enclosed, the nuclear material is not enclosed by a nuclear membrane, and the nuclear material will be called as a nucleoid or a genophore or an incipient nucleus, and the nuclear material will be called as incipient nucleus or nucleoid. However, in the eukaryotic cell we will see that the nuclear material will be enclosed by a proper nuclear membrane, presence of nuclear membrane, right. The organelle, the cellular organelles over here will be absent. The complex, or rather than writing the complex we should write the membrane bound organelles, the membrane bound organelles will be absent, and here the membrane bound organelles will be present. The membrane bound organelles like Golgi body, endoplasmic reticulum, etc., will be present. Then after the membrane bound organelles we can talk about the next thing, that is the prokaryotic cell. So the membrane bound organelles are absent, and the membrane bound organelles like Golgi body, etc., will be present. Now ribosomes and all will be present. Over here the ribosome will be 16S type of ribosome, and over here the 18S type of ribosome will be found. 18S type of ribosome, right. The prokaryotes, again, they can be of various sizes. Even the eukaryote can be of different sizes and different multicellularity. The prokaryotes will be usually, these are single cell, they can be single to many celled. These are single celled, these are single celled, these can be single to many celled. These can be single to many celled, and that is the difference between prokaryotic and eukaryotic cells, right. Now we move on to the next question. What are mycoplasmas? The mycoplasma are also called as mycoplasmas are also called as pleuropneumonia like organisms, as pleuropneumonia like organisms. PPLO. These are pleomorphic, that is they can change their shape. These are pleomorphic, that is they can change their shape, okay, that is they can change their shape. These are prokaryotes, right? These are prokaryotes and lack the cell wall, and lack the cell wall, right? This is the main thing, they lack the cell wall. They are also called as pleuropneumonia like organisms. They can change the shape and hence they are pleomorphic in nature. Friends, for this, we have come to the end of this discussion. In the next session, we will talk about something more interesting. Take care of your health, and the time we meet again, a very goodbye from this side. Thank you.